This is the first conference of the cycle of conferences included in the Campus do Mar. We have here a group of people belonging to the Master of Oceanography of University of Vigo. There are students, there are teachers, but there are also other people in the main building. And I think this is going to be the conference. This is the place where we can start making questions after the conference, but it will be also some questions coming from the other groups. Now just have to say that it is a pleasure for me to introduce today to Dr. Deborah Iglesias Rodriguez. She works since 2003 in the National Oceanography Center in Southampton, but she started her studies at University of Santiago de Compostela and did her PhD in phytoplankton carbon physiology at the University of Wales in the United Kingdom. She is very well known worldwide for her work in phytoplankton physiology, mainly in ocean acidification, but also in other topics as proteomics and genetics of phytoplankton. Has a very long list of publications and reviews, professional contributions, very, very long and extensive CV. But especially relevant is, especially for the topic of today's conference, is her paper, Phytoplankton Calcification in a High CO₂ World, published in Science in 2008, that was identified by Thomson Reuters as the fast-breaking paper and one of the top most cited papers in the field of geosciences. So I'm sure that it's going to be a very interesting conference. So when you want, go ahead.
Thank you very much. It's a great honor to be here as part of this conference series. And I'd like to thank Emilio Fernandez for bringing me here to the series. And I wish him luck in this really exciting initiative and project of Campus do Mar. So this is a wonderful thing for Vigo, and I hope that scientists, technologists, and entrepreneurs benefit from such a great initiative. So actually Vigo is my alma mater. I started studying in Vigo in the Colegio Universitario de Vigo, and then I graduated at Santiago de Compostela. So this is a wonderful thing. So yeah, it's great to be here. So yes, the topic is of my talk today is how marine biota, how marine life adapt to a high CO₂ environment. And I have in this first slide a number of organisms from single cell plants called phytoplankton up to sea urchins and corals and so on. So I focus on marine phytoplankton and also sea urchin larvae. So we have started doing some work on the development of marine larvae. And so I work in the National Oceanography Center in Southampton, and I do a combination of lab work with field studies. So here you see the building and the one of the big NERC ships, the James Cook, and this is some of the work I do in the lab. Some of my students, we go to sea and we work in the lab as well. And these are some of the people who contributed to the work I'm presenting today. So this is from my lab in Southampton. And as I said, I come from Vigo, so I just decided to show some images from Vigo. So this is at the top, me and my little brother Pablo, who's now a grown-up man, and then on the left we have some images from around the Rio of Vigo, near Cabone and so on. So I've been very connected to the sea and since I was little, and then I became an oceanographer, and I'm very fortunate. I often think I have the best job in the world because I get to see really amazing places. And I just put together some images from a recent trip just 4 months ago to the Arctic. And the little picture at the top is a penguin, of course, not from the Arctic, from Antarctica. But what I'm trying to say is that my job takes me to really amazing places, and I have the opportunity to do very exciting work. I tend to go to cold places because the solubility of CO₂ increases with decreasing temperature. So these are really interesting places to test the effect of ocean acidification. It's kind of like an analog for testing ocean acidification. And of course, we get to see this amazing wildlife. So these are pictures that I took in the Arctic last October as part of a Cape Farewell expedition. And I started working with these animals called seawater butterflies or theropods. And I'll show you a little movie a little bit later on. So the oceans are incredibly complex and the oceans are changing. So I am working on a tiny little part of this equation, which is how CO2 impacts upon phytoplankton, primary production, and other functions like calcification. But in fact, there are these reciprocal interactions between CO2 and temperature and oxygen, and they all affect marine animals and plants in many ways. So it's extremely difficult to pay attention to all these factors that are going to impact upon what we are measuring, but also the oceans work in very different time scales and spatial scales. They are an incredibly dynamic system. So it is a challenge to study these questions. So this is probably the best shown, the most popular graph when one talks about ocean acidification. So here you can see the trend in red of CO2 in partial pressure from the Mauna Loa Observatory. So this is in the atmosphere. And if you see the partial pressure of CO2 in seawater, it kind of mirrors what we see in the atmosphere. So this is this trend here. I have to show you the trends with the mouse because, yeah, I cannot use a pointer, a laser pointer here. So this is the CO2 in blue from the Mauna Loa, from the Aloha Observatory in the seawater. So you can see that it goes in parallel with the PCO2 levels in air. And if you measure the pH of the water at the same station, you can see that it is inversely proportional to the changes in CO2. Yeah, so you can see that here pH declines as CO2 increases in seawater. So we know that in most places in the world, these are the changes that are taking place. There are exceptions, like in the North Pacific, in the Bering Sea, but generally the oceans are experiencing an increase in carbon dioxide levels, which are happening in parallel to the changes in the atmospheric PCO2 levels. So the central issues in the community working on ocean acidification are how is ocean acidification affecting functions in marine life, like photosynthesis and calcification, nitrogen fixation, silicification, and so on. What are the combined effects of other variables, like temperature, for example, or like oxygen? I mean, we are focused on CO2 in these kinds of studies, but actually an increase in temperature will result in a decline in the oxygen solubility as well. So these are very important parameters when we are talking about animals and plants. Everything respires. All animals and plants respire. And then we are studying photosynthetic organisms that are, whose health and functions are critically dependent upon the CO2 availability in the media. Another question is what controls the responses to ocean acidification? In other words, what is the versatility of organisms? How do organisms adapt in the long term and acclimate in the short term to these changes in the chemistry of the oceans? And is it triggered, is it controlled by genetics? Is it controlled by physiological adaptations in the short term? Or is it, are there other controls? And then of course there is this, threshold values or what are called tipping points. So which are the levels beyond which the organism moves into an irreversible state. And ultimately the socio-economic impacts of ocean acidification. And I show here an example on the potential effect of a decline in theropod populations on fish stocks. So this is part of the reason why I went to the Arctic a few months ago. And I went to study these organisms called theropods. So these are the really susceptible regions to ocean acidification. So this is part of the reason why we are studying them. Now, studying ocean acidification, the chemistry of ocean acidification is incredibly complex because it's not linear. So we have these changes in CO2 and bicarbonate and carbonate ions, but these changes do not change, these changes do not happen in a linear way. And the range of values that we are expecting to happen in the near future will be going into this direction, to the left in terms of pH. So this is the range of values from what we are seeing today to what we may see in the near future. So essentially what this means is that there will be an increase and a decrease in some of these variables. So essentially, as CO2 enters seawater, it combines with water and it forms carbonic acid, which is a weak acid. And therefore, the term ocean acidification refers to a decline in the pH of the oceans. The pH is still alkaline, but there is an increase in protons in the seawater. So there are some chemical changes happening because of that. So the first thing that happens is that the concentration of carbon dioxide increases in seawater. The same takes place with the concentration of bicarbonate ions in seawater. Now, these two molecules can be used in the process of photosynthesis. And bicarbonate can be an alternative source in the process of calcification. And I want you to remember this because this will be relevant a little bit later on. Now, what happens with as CO2 enters the ocean is that carbonate ions decline. And this is relevant to organisms that calcify because that's the primary source of carbon for calcification. Yep. And there is a term that is broadly used in ocean acidification, which is this omega value, or the saturation state of calcium carbonate, which is critically dependent upon the concentration of carbonate ions and the concentration of calcium in the seawater. Now, because the concentration of calcium in seawater is semi-conservative, as carbonate ions decline, this will have an effect on omega. And of course, pH has an effect itself on the physiology of organisms. So, why do we care about this? And we are looking at biological functions as well as the changes in the carbonate chemistry. So we are, the community working on ocean acidification is interested in photosynthesis, nitrogen fixation, calcification, reproduction, and so on. So these are some of the things that some of the processes that I have reviewed recently to try and come up with the state of our understanding on ocean acidification. And the answer is that we still need to know a lot about the effect on some of these processes. So why is pH important in organisms? Well, pH affects substrate speciation, many physiological processes, enzyme activity, so the catalytic properties of enzymes, of proteins, and crystal size, mineralogy, crystallography and structure. So we also need to think about the fact that we often think of cells as having a pH inside the cell similar to the pH outside in the seawater, but that is not true. If we consider an average pH for open ocean seawater of about 8.1, and if we think about the pH inside the cells being between 7.1 and 7.8, we can see we can imagine that there are processes that are going to be detrimentally affected by ocean acidification, but there are others which are energetically more advantageous in the presence of higher CO2. So this is for intracellular compartments. So inside the cytosol of the cells, these are the typical pH values between 7.1 and 7.8. However, in the calcification reservoir of all organisms, they have to increase the pH. So it has to be, the pH is typically above 8. So they have to increase the pH, and for that, they need energy. So they need to be able to extrude protons to outside the calcium carbonate reservoir. So metabolism itself produces and consumes protons. So the cells have to have relatively sophisticated ways of extruding and bringing ions in and out of reservoirs. So this is what we all are trying to figure out in corals, in coccolithophores, and in other organisms. So to be able to determine or to figure out which are going to be the winners, if you like, and the losers in a high CO2 world. And of course, for this, we try to think about the best experimental approach. So there are many experimental approaches. There is no perfect approach, but it's very important to try and reproduce the carbonate chemistry in the best possible way experimentally. So this is just an example, and I won't go into detail, but if you think about this, the inside of the cell, and specifically inside the cell of the calcium carbonate reservoir as compartments, and consider the seawater as another compartment. We have all these processes that require, some of them require energy. So you can see here that protons are going in and out of the cell. And then we have the calcification reservoir, and you can see that the formation of calcium carbonate here at the calcification reservoir actually generates carbon dioxide. So this is a really interesting thing, that calcification is considered the largest carbon sink on Earth on timescales of millennia. So on long term, in the long term, calcification is the largest carbon reservoir on Earth. But in the short term, the actual process of calcification generates carbon dioxide. So for a while, it was thought that this production of carbon dioxide could be actually a CO₂ concentrating mechanism. So a way for the cells to acquire CO₂ for the site of Rubisco and improve the photosynthetic rate, photosynthetic carbon fixation. There is evidence supporting and against this hypothesis. So this still remains an open question. But there are all these really interesting, if you like, reciprocal interactions between processes in and out of each of these reservoirs. So in some ways, the challenge is to be able to tease them apart and to understand what are the sources, for example, of carbon for calcification. One very important, something that I wanted to point out from this slide is the fact that bicarbonate, the interconversion of carbonate to bicarbonate is extremely fast, but in a high bicarbonate world, if you like, the organisms that can use bicarbonate efficiently by converting that into carbonate are going to be okay, essentially. So they are going to be able to deal with the elevation in CO₂, the elevation in bicarbonate, but at the decline in carbonate ions. So this is a very important question, and I think it lies at the, really at the having this answer is going to be incredibly, is going to shed light into why some organisms are doing okay or even better in high CO₂ environments. So how do we go about it? Well, there are many different experimental analogs for ocean acidification, and as I said, there is no perfect analog, but so we can work in the lab, we can work in the lab in small scale or in large scale, we can go to in between the lab and the field and do mesocosm experiments, and we can do tests in the field. And we can also take advantage of areas that we call hotspots, so areas that are already experiencing ocean acidification. So we can go there and we can test how organisms are doing there. We can test the morphology of the shells, the quality of the shells, and the dissolution, the potential corrosion of the shells, and how they are actually, whether they are growing in comparable numbers to adjacent waters and so on. So I'm going to show you some examples of how the work we are doing. And primarily I work on phytoplankton. So here, I just showed you this movie of dinoflagellates. They are actually having sex right here. We film them. We can actually induce gametogenesis in culture. So it's a very rare sighting for us and quite an exciting one. But the Millania hexilii is the organism that I'm going to focus on now. So these are beautiful calcium carbonate producing organisms. They form these huge scale blooms. They are infected by viruses at the end of blooms. And when that happens, they actually shed the shells of calcium carbonate. The cells break down, and all this organic and inorganic material starts the inevitable downward flux to the deep sea sediments. So this is a really important organism because it also affects the sulfur cycling. As you probably know, it produces this cloud condensation nuclei from this gas called dimethyl sulfide. So they affect the chemistry and potentially the biology, and we can see the optical signal from satellites. So this is a very nice organism, model organism to work with, because not only you can work in the lab, in a test tube with it, but you can look at the distribution in space and time using satellite technology. So there are two main questions that I am interested in. One is that of acclimation, so the short-term responses to, in this case, ocean acidification. So how do coccolithophores or other organisms respond to these short-term changes in carbon dioxide and in the carbonate chemistry associated with ocean acidification? And then the other question is, how do they adapt? What is the selection happening within the population and within communities? What is the long-term trend of adaptation? How do organisms adapt in the long term to environmental selection pressure? So for this reason, we have to really understand what's going on within the cell. So, here I have a cartoon of what a typical coccolithophore cell looks like. So we, as I said before, we cannot ignore other processes that are happening together with calcification. And the key thing in my view, for understanding what organisms or what groups of organisms are going to adapt well to ocean acidification is to really try to figure out which organisms can use bicarbonate efficiently and which are dependent exclusively upon carbonate or largely upon the availability of carbonate ions. So, and really, if we can understand this, if we have an experimental approach that will give us that answer, I think we will make a great progress in understanding the effect of ocean acidification on biota. So what we know so far is that photosynthesis either increases or is unaffected by ocean acidification. And this kind of makes sense because you put more CO₂ in the water, you increase the substrate of carbon for photosynthesis, and this is what we generally find. And this is found in diatoms and is found in coccolithophores. We need to also understand the synergistic effects of temperature and CO₂ and other factors like oxygen, for example. So we have evidence that if we conduct, for example, multifactorial experiments and we add temperature as another variable, not only the magnitude but the direction of the response can be very different from just using CO₂ by itself. And there is evidence that ocean acidification can alter the availability of metals, and pH can also shift nutrient stoichiometry. And this can have an effect on the community structure and the relationships between different trophic levels. So a very interesting thing is, for example, just thinking about an increase in carbon in the system will increase the carbon to nitrogen ratio. Well, larvae, zooplankton depend on that biochemical, nutritious, if you like, quality of these plankton particles. And if you alter that, the effect on larval health remains to be tested. So, so what about calcification? So the story with calcification is a little more complicated. Well, so I've just showed you the effect on photosynthesis, the effect on nutrient stoichiometry, and so on. Now with calcification it's a little trickier. So I will start with defining calcification. Well, everything calcifies more or less. So we have, from mammalian placenta calcification to, the formation of spicules in echinoderms and coccolithophore calcification. Most phyla calcify. So there are many factors that affect calcification, and this is another tricky thing that, this includes nutrients. For example, phosphate and sulfate inhibit the formation of calcium carbonate crystals just by themselves. And then there are other metabolic effects on calcification. And there are, we work with animals and plants that make a large contribution to the global calcium carbonate budget. And recently there was this work by Wilson et al. That showed this contribution in fish, for example. And of course, coccolithophores play a big role in the global calcium carbonate budget. So there are two main elements that we focus on, and one is this, essentially the source of carbon for calcification, carbonate and bicarbonate ions. And there are many different experimental approaches, and I apologize about the size of the font here, the font size, which is tiny, but I wanted to have all these figures within this slide to compare. And I just wanted to show you, on the left, we have two species of coccolithophores. Here, Calcidiscus leptoporus and Coccolithus pelagicus. So these are two species typical of subtropical ecosystems. And what I wanted to show you is the apparent malformation of the coccoliths at in the top case in the Calcidiscus leptoporus with high and low CO2, both high and low CO2. So they appear to have a non-uniform response to the effect of ocean acidification, and this is work by Gerald Langer and others in 2006. In the case of Coccolithus pelagicus, the response is not so clear either, but there appears to be malformations with increasing CO2. More recently, Gerald Langer and others in 2009, they published this work on the differences between strains of what we call the same species of Coccolithophorus, between strains of Emiliania haxillii. So I just showed you differences or responses between species, but in fact, if you look at within the species concept, at different clones, different strains, their response can be quite different as well. Then I wanted to show you another study, or two studies working with Emiliania haxillii, and in this case, this is the same strain of Emiliania haxillii, this is the work that I published in 2008, in which I show an increase in calcification, represented here by the increase in the mean coccolith size, in white, and here in black you can see the inorganic carbon or calcium carbonate per cell in these cultures maintain under different CO2 partial pressures. And this was achieved with bubbling the water with different CO2 partial pressures. So essentially you buy air which contains different partial pressures of CO2. So we used just different values for that. And we found also an increase in the size of the coccoliths and as well as an increase in the size of the cells. So this was really very different from the trend, from the previous trend that we had, that we knew until then. One thing that I wanted to point out is that these omega calcite values that you can see here in red, they all go down to 2.6, and the threshold value below which it is expected that calcium carbonate may dissolve is 1. So they were above that threshold value, but we pushed the system, if you like, up to 750 parts per million of CO2. Yuan and Morel, a group from Princeton, they did the same, using the same strain, and they found the same trend in terms of the response to ocean acidification. So they found an increase in the calcification, standing stock per cell, and also the production rates in mass per cell per day. And if we compare this with previous studies by Ulf Rübezahl and others, we find that I'm just showing you a snapshot of the results here, but they found a decline in calcification, and I illustrate that here with two species, with Emiliania haxillii here, which is the same as I used. So this is present day conditions, and this is elevated CO2 conditions. And they found malformations under elevated CO2 conditions in both Emiliania haxillii and also Gypharocaps oceanica. Yeah. So now there have been a little bit of discussion about why we found these differences in the results. And initially, some of the discussion revolved around the methodology. So in previous studies, maybe 50 years of phycological research involved adding acid to the cultures or manipulating the carbonate chemistry with using acid. But in our case, we use this other method, and the reason for that is that if one manipulates the media with acid, you essentially you do not change the bicarbonate levels in the media. So we felt that we needed to reproduce in a more realistic way the carbonate chemistry that we should expect in future high CO2 waters. So yes, I wrote a review a few months ago, and I tried to bring together the state of our knowledge on the effect of ocean acidification on marine biota. So here you have at the top the response to increasing CO2 levels from decline, increase, non-response, and non-uniform response. And you can see that there are, at the time when I wrote this, which was, this was published a few months ago, maybe 3 months ago, 4 months ago, there are, there is significant variation for all, for most organisms, actually for all groups that I am presenting here. And since then, there has been a huge amount of work. I mean, these are previous pieces of work and future pieces of work, but there is a huge body of work that is trying to figure out what lies behind these responses. So, and there is emerging work that you are going to see in the next few months. So what are the biogeochemical implications? So apart from the ecological responses, the ecological implications, there are biogeochemical effects of all this. And one very important one is this ratio between the inorganic and the organic carbon, the solid phase inorganic to organic carbon ratio. Yeah. And it is considered that if this ratio is above 1.6, coccolithophores or whichever calcifiers you are working on represent a source of CO2 to the surrounding waters. And if this ratio is below 1.6, then it represents a sink of CO2 to the surrounding waters. So we have, this is a really important factor to take into account. In previous studies by Ribesel and others, they found a decline in this ratio. Yeah. Further studies by the same group found a non-uniform response, and what we found was a neutral effect with increasing CO2 levels. And here I just show you on the left the effect of increasing inorganic carbon on photosynthetic carbon fixation in diatoms, as an example in non-calcifying organisms. So generally, for organic carbon production, the effect of ocean acidification is that of increasing photosynthetic activity, or in very few cases, neutral effect. So there has been a lot of discussion about where the uncertainties actually come from. So one possibility is that there may be huge genetic variability between and within the species concept that is behind these different responses. I think that that may be the case, but it may also be the fact that in some of these organisms, the ability for them to convert bicarbonate ions into carbonate ions and then use carbonate ions in calcification may be the reason behind them, their acclimation, if you like, or potential adaptation to increasing PCO2 and decreasing carbonate levels. Now, in terms of the way we do the experiments, and there is no perfect experimental design, but we are running very short-term, abrupt manipulations, and the system doesn't really work like that. So typically we have a culture in the lab, and we just expose this culture to very sudden changes in the carbonate chemistry, or in temperature, or in whatever we are testing. So there are other really interesting approaches that we can use, and they are relatively novel in the phycological work, like slow, evolving experimental manipulations. So in the case of coccolithophores, there is only one midterm incubation, but yeah, the carbon chemistry was manipulated using acids, so the bicarbonate levels were not reproducing the future conditions, and there is no measurement for molecular evolution. So this is really important because in the different, we need to build models for calcification, and here I'm just showing you 3 potential models for calcification, and this is work that I just submitted. So corals have the calcification reservoir more isolated from the external medium than coccolithophores, for example. Sorry, more in contact with the external medium than coccolithophores. So in the case of coccolithophores, like you can see at the bottom here, they form the calcium carbonate in this coccolith vesicle, which is completely isolated from the external medium. They are able to wrap these coccoliths, if you like, in an organic film that may act as additional protection for the mineral once it is produced. And in the case of foraminifera here in the middle, they form this vesicle where they control the pH of this vesicle by extruding protons, so they maintain the pH alkaline, and they move this vesicle towards the building blocks for calcium carbonate in the animal. So there are different ways, different adaptations to of calcification and different mechanisms. So this is what we are aiming for, to really come up with models that can help us. The problem we have is that not only when one looks at different species or different strains within species, but an additional problem is if one looks at changes during the life cycle. So when we go out there and we sample, we have to remember that there are changes in the biochemical imprint of organisms. So for example, if you think of a bloom developing, we typically see a pretty stable peak-to-POC ratio. Yeah, so the cells are growing exponentially, and the inorganic to organic carbon ratio, let's say, is 1:1, for the sake of the argument. Then the cells reach the stationary phase here, they run out of nutrients, and calcification rates per cell are higher, not because they calcify more, but because they photosynthesize less. So this peak-to-POC ratio changes. And at the end of the bloom, the cells, the populations appear to form gametes, and gametes do not carry any calcites. Yeah. So the biochemical characteristics of the different life cycle of organisms can vary. And if we look at the different phenotypes within populations, within the species concept, we can see that they are highly variable as well. So if you're a modeler, which one do you use? And I'm showing you a naked strain of coccolithophores here, and 2 strains. And in the case of this one on the right, it calcifies 10 times faster than the middle one, than the one in the middle. So, if you are a modeler, you have very different functions associated with what we call the same species. So this is a big, big challenge. So we have to think of creative ways of testing this. So one thing we did is this summer we went to the PAP site, the Porcupine Abyssal Plain, because it has very deep waters, and we brought our actual cultures there. Typically you go and target populations. Unfortunately, there are no coccolithophore populations. So this was our first testing of looking at the effect of deep waters. And I'm just showing this as an alternative to working in the lab with artificial conditions. So the test was how does an upwelling event, and these are on the increase, the frequency of upwelling events is increasing, and we actually don't know the physiological effect on cells. So how do cold, high CO2 waters affect the physiology of coccolithophores? Yeah. And specifically, how does low carbonate ions affect calcification in coccolithophores? So this is very, very recent. We are still analyzing the samples, but I just wanted to show you this approach because I think it's very interesting. And some of you are doing this kind of work, and there are many places that one can target to do these kinds of studies. And what we have already completed the analysis for echinoderms. So we took sea urchin larvae of some Echinus miliaris, and we went to the same site, and we found that there was no effect on development, but there was effect on the formation of the spicules, which are essentially calcified structures. It's kind of like the skeleton of the larvae of echinoderms. And there was also a decline by 68% in the fertilization success in larvae. So we know that there is some effect on larvae, but the effect on development appeared to be neutral. So we followed the same model, and we went back there with our coccolithophore cultures, and we collected water from 4,780 meters approximately, from 1,000 meters, 500 meters, and 100 meters. And then we ran our experiments, and we're in the process of analyzing the data. But essentially, we found no correlation between the cell size and the coccolith size in both strains, although the coccoliths appear to be more damaged at 500 meters. We are actually analyzing the samples right now, and we use 2 morphotypes. This is another morphotype of another strain of Emiliania haxelii, and again we didn't find. These are the cells maintained in water from the chlorophyll max depth, which was around between 30 and 40 meters, 500 meters, 1,000 meters, and 5,000 meters. So that we didn't find any clear differences, although the cells some of the cells, some of the cultures maintained in deep waters appear to have larger cells, but we didn't find a straightforward correlation. Now, the interesting thing that we found is that for all conditions and in all cultures, the peak-to-POC ratio appeared to be above 1.6. And this is something that we are now looking at, because the implications of these are that for these particular conditions, these organisms are actually a source of CO2 to the surrounding water. So we are actually looking at what this means right now. So this is one potential approach to try to understand what happens in the real environment. But there are evolutionary questions about how organisms do in the long term. So how does a change, for example, in the elemental ratios affect the biochemical composition of phytoplankton? And how does that affect grazing? How does that affect zooplankton population and larvae of fish or echinoderms and so on? Also, the effect on size, we found bigger cells. We found a decline in the growth rates with increasing CO2, but we found bigger cells. So how does that affect grazing by zooplankton? And then there is the really key question of how do calcifiers versus silicifiers versus nitrogen fixers are going to be like? What are the proportional, what is the proportional representation of these functional groups in the future? And this is very important because they control the cycling of elements between the atmosphere, the upper ocean, and the ocean interior. And of course, how fast do organisms adapt to these changes? And what we are finding now is that the levels of CO2, for example, that we are experiencing and the decline in carbonate ions are not unprecedented in the history of Earth, but these changes are the fastest that the Earth has experienced for perhaps the last 55 million years. So we are trying to find out whether and to what extent animals and plants are going to be able to adapt to these changes. And this is what I was trying to talk about before, that we have these different temporal and spatial scales, and it's very difficult to bring them together and to try and make predictions based on what we see from the geological record, for example. So we have examples of past periods of ocean acidification, and we can see that, for example, in the Cretaceous, coccolithophores were well represented in the geological record. However, it's very difficult to uncouple the effect of elevated CO2 from other effects. Yeah, so in the Cretaceous, the alkalinity was different. Calcium levels were about between 2 and 3 times higher than today. So it's very difficult to tease apart the distinct effects of CO2 versus other environmental variables. So we have now what we call the new omics approach. So these are technologies that go from genomics through to transcriptomics and to proteomics. And they allow us to look at the diversity, the potential in terms of the genes that organisms carry. So it's kind of like the Dawkins way of thinking about organisms as mere gene carriers with potential functions. But one thing that's, we have the genomic level, which is pretty well established for field studies, and then we have the transcriptomics level and the proteomics level. So with the proteomics level, the nice thing is that one can look at what organisms are doing potentially. Yeah. So while genomics tells you what is there, proteomics can potentially tell you what they are doing. So for a biological oceanographer working on processes, is a very attractive approach. And therefore, if you have an organism that divides very fast, like coccolithophores, like some species of coccolithophores, you can run experiments where when they undergo many generations, and you can actually slowly have incremental or decreasing pulses of carbon dioxide, and look at how organisms adapt in real time. So you can look at how the genome and the proteome and the transcriptome changes over time. So I mean, this is a very these are very early days, but this really is we have the technology to do this. And I just wanted to show you this because this is very nice work by Peter Bondasso, and what you can see in the cartoon on the left is the intersection of the common express sequence tags identified in different strains of Emiliania huxleyi. So each of these enclosures, if you like, represents one strain of Emiliania huxleyi, and they have some common express sequence tags. In other words, they express some of the genes all these strains express a subset of their genes. Now, if you look at this graph on the right, and you can see that there is great variability in the transcriptome at the transcriptomics level. But if you move to the right, what Peter Bondasso and colleagues represented here are the EST, the transcripts, the expressed genes that are unique to haploid cells, in other words, to gametes, to the sexual cells, and the transcripts that are unique to the asexual or vegetative cells. And you can see the difference between the vegetative and the gametes in this case. So you can see that about 50% of the genes expressed in the gametes are not expressed in the vegetative cells. Yeah. So this is a really interesting piece of information, because it tells you that even within the same cell, when it undergoes different stages of its life cycle, it will express different functions. Yeah. So this is really, it's a beautiful piece of work, and a starting point for a lot of people to start thinking about, well, how can we figure this out, when we go to the field. So what we decided to do is, we have genomes of organisms that represent the potential for different functions. So, they give you an idea of the diversity of genes that they comprise. What we did is go one level beyond and look at the proteome. So essentially sequence the proteome of, in this case, strain of Emiliania haxillii. So we produced the first marine algal, eukaryotic algal proteome. And this, while the genome, working at the genome level, will probably be quite limited in terms of telling you about the different functions of cells, the proteome is giving you details at the most fundamental level of biological organization, which is the proteome level. And to do this in the field is currently not possible. We have this challenge of having to optimize the technique to be able to apply that to the field. But it's an extremely exciting, we are looking forward to continue working on this and hopefully be able to develop something for the field. So what we are doing in the lab is looking at CO₂, doing CO₂ incubation experiments and try to sequence a proteome, a low CO₂ proteome and a high CO₂ proteome, if you like, to look at what is associated with low and high CO₂. And finally, I just wanted to end with a little bit of a, very different thing. So the knowledge of what I was talking about today, so the effect of ocean acidification and how organisms respond and biological functions and so on. This information can be used to design geoengineering approaches. So something that I was at the Asilomar meeting in California, in Monterey, in Asilomar, and there have been many discussions about ways to, for example, sequester carbon, sequester CO₂ in the oceans, and of course, solar radiation management and many other applications, potential applications. But in the case of sequestering CO₂, one could think of using this information to really devise a scheme where one could use the knowledge on the metabolic pathways of some of these organisms to try and enhance some of this carbon sequestration. So we know what these cells do in terms of producing and consuming CO₂, and perhaps this could be there could be some potential for using this information in geoengineering approaches. And finally, there is this idea of forming this synthetic or artificial chromosomes, and this was put forward by Craig Venter, who actually designed the first artificial chromosome. So one can have these designer functions, if you like, in microorganisms to try and enhance and functions or repress some specific functions. So this is early days, and of course the deployment in the field is not possible and is probably not desirable. But this is something that one should keep in mind, because the technology is there to be able to try to counteract some of the problems that humans have caused in the environment. And just the final, I think it's the final slide, I just wanted to tell you that there are there is an international effort to maintain, to improve, and to continue with the long-term observation of changes in environmental variables. And ocean acidification is now very much contemplated in these observatories. So there is an international effort in monitoring ocean acidification in the field. And the stations that you can see here in red are ocean acidification monitoring stations in general, and the yellow ones are for coral reefs. So there are plans for looking at, for doing long-term manipulation experiments, and there is, as I said, an international effort to use the same techniques, the same approaches, and to really have a consolidated data acquisition for what's going on in the oceans in terms of the chemistry and the biology. So in summary, ocean acidification, we know that it enhances generally photosynthetic carbon fixation, but the calcification responses are not so clear. There is an urgent need to elucidate these responses in calcification. In some organisms, they appear to be relatively clear, but in the case of coccolithophores, they really remain an open question at the moment. It's very important to keep in mind the synergistic effects with other environmental variables like temperature and so on. And in going from the lab to the field, it's very desirable to think of smart experiments that try to reproduce evolution, if you like, in a more realistic way. And these are some of my co-authors and people from the National Oceanography Center and collaborators who had something to do with the work I presented here. And I'd like to thank you all for your attention. Thank you.
Thank you very much.